Good morning. It's December 12th, and this is your daily brief in science. Here's everything you need to know. Lilly's retatrutide delivered strong results in a Phase 3 obesity trial for adults with knee osteoarthritis, achieving an average 28.7% weight loss at the 12 mg dose and a 4.4-point improvement in WOMAC pain by week 68. The trial met its co-primary endpoints, showing significant reductions in body weight and WOMAC pain from baseline to week 68. Nearly half of participants on retatrutide reached at least 25% weight loss, with meaningful shares surpassing 30% or 35%, far outperforming placebo. Lilly plans to release additional Phase 3 results over the coming year and pursue regulatory submissions, signaling a relatively straightforward path barring safety concerns. The company announced the TRIUMPH-4 results in a statement, noting 5 dose levels and more than 5,800 participants across studies since 2023, with details to come at future meetings and in peer-reviewed journals. Administration is via weekly injections, and FDA submission is planned after 7 more Phase 3 trials in 2026, with potential approval possibly in late 2026 or later depending on the review. The trial's positive data comes as the global weight loss drug market is projected to grow substantially, around $100 to $150 billion by the mid-2030s, highlighting retatrutide's strategic importance. Side effects include nausea and stomach issues, with risks of gallbladder and pancreatic disease. Patients should discuss individual risk profiles with their doctors. The report is a Stat Plus exclusive and requires a subscription for full access, and market sentiment around the stock has been mixed amid bearish chatter on Stocktwits, despite a year-over-year gain. Retatrutide could help Lilly defend obesity and diabetes drug market share as competitors pursue similar therapies. The drug's administration and regulatory timeline frame the near-term milestones for its development. 2 top predators, killer whales and dolphins, were observed in British Columbia's coastal waters, sharing a hunting moment where killer whales trailed the dolphins and fed on salmon pieces, while the dolphins appeared to exploit the prey. In August 2020, 9 northern resident orcas were studied using movement data, underwater video, acoustic recordings, and aerial drone observations. Researchers logged 258 unique dolphin-orca interactions and described the behavior as a collaborative foraging partnership through underwater tags, satellite data, and drone footage. The study, a multi-institution collaboration led by Dalhousie University marine scientist Sarah Fortune, involved the University of British Columbia, the Leibniz Institute, and the Hakai Institute. Fortune and colleagues characterize this as behavioral coordination within the northern resident population, though it does not meet the stricter criteria for mutualism where both species benefit. The findings suggest possible cooperation or coordinated hunting, with dolphins taking advantage of the predator's presence to access prey. Experts say more research is needed to determine how consistent these interactions are, what drives them, and what ecological significance they may have for both predator groups. The research underscores the need to update understanding of marine mammal behavior in light of climate-related changes to the British Columbia coast. The study was published in Scientific Reports on December 11, 2025, and highlights a collaborative approach to observing interspecies foraging. It also raises broader questions about interspecies cooperation in marine foraging and its ecological implications, including how changing ocean conditions and prey availability may affect social and foraging behaviors. More observation is needed to assess the prevalence, stability, and ecological impact of these interactions along coastal British Columbia. A Penn State-led study shows wood warblers acquire color diversity not just from their own relatives, but through gene sharing with evolutionary neighbors. This horizontal transfer happens mostly via natural hybrids that backcross with parental species, letting color genes move beyond a single lineage. Researchers analyzed DNA from 400 warblers across 100 species, focusing on 3 carotenoid-related genes to trace movement and comparing gene trees with the species tree. Dating and directionality point to introgression from Laeothlipis, roughly 500,000 to 2 million years ago, with more recent ongoing gene flow into red-faced warblers. The work appears in PLOS Biology on December 11, 2025, led by David Tooze and colleagues at Penn State, with NSF and Penn State funding and collaborators including Lan Nai Pung, Andrew Wood, Marcella Bays, and Irby Levette. This pattern of cross-species gene transfer aligns with findings in other groups and suggests a broader evolutionary principle worth deeper study. Looking ahead, researchers anticipate more cross-species gene sharing as genome data accumulate, which could reshape how we view evolutionary history. Findings imply substantial crosstalk between lineages, with hybrids acting as conduits for beneficial mutations and complex trait development across species boundaries. Ongoing work includes sampling more red-faced warblers to track introgression spread and ecological impacts, plus planned functional studies on the borrowed BCO2 variant. Evidence points to more recent introgression into red-faced warblers and geographic spread, and ecological effects are being studied as sampling continues. Introgression of the BCO2 gene occurred across multiple lineages, including transfers from Laeothlypis to Setophaga and Cardellina, and from Vermivora to Geothlypis, with timing spanning hundreds of thousands to millions of years. Some transfers date from about 500,000 to 2 million years ago, and the borrowed gene persisted through subsequent speciation. A new approach targeting UPAR may renew aging gut tissue. Researchers at Cold Spring Harbor Laboratory report that anti-UPAR CAR T-cell therapy can stimulate intestinal cell growth and repair, improving gut health in aging and radiation-damaged mouse models. In young mice, a single anti-UPAR CAR T-cell treatment produced lasting effects, with UPAR fighting cells persisting for at least 15 months and continued reductions in cellular senescence alongside better gut health. Led by Karina Amor-Vegas, Samir Bayaz, and Onur Eskiokak, the study delivered anti-UPAR CAR T-cells to the intestines of both old and young mice, yielding significant gains in nutrient absorption, decreased inflammation, and faster epithelial regeneration after injury. Funding for the research came from multiple institutions, including the National Cancer Institute, NIH, the Mark Foundation, Memorial Sloan Kettering, Chan Zuckerberg Initiative, and others. When compared with senolytics like dasatinib and quercetin, the approach similarly reduced senescent cells and helped restore stem cell function, suggesting convergent benefits across strategies. Safety notes: treated mice did not develop intestinal cancer, but authors urge clinical trials to evaluate safety and efficacy in humans. In older, 18 to 20 months, and young mice, UPAR-expressing cells are more prevalent and co-express senescence markers, with older human gut tissue showing a similar pattern. Conclusion: UPAR-positive epithelial cells are proposed as key drivers of intestinal aging and inflammation, presenting a promising therapeutic target pending human trials and safety assessments. UPAR-positive senescent cells are harmful when abundant, and prior work showed CAR T-cells targeting UPAR can reduce senescence in other tissues. Background on aging and gut health: aging impairs intestinal epithelium regeneration, contributing to inflammaging and leaky gut, often worsened by cancer radiation. The gut epithelium renews rapidly but declines with age, leading to leaky gut, inflammation, and accumulation of senescent cells. Functional restoration observed in aged mice included improved organoid-forming capacity, reduced inflammation and dysbiosis, and a gut microbiota profile closer to that of younger animals. A new study published in Science Advances tackles a century of pollution by looking at bones from 94 individuals in 2 English towns, one industrial and one rural. The researchers combine historical records with bone chemistry and isotopic analysis to measure lifetime exposure to heavy metals. The work challenges a simple urban versus rural narrative by showing that health impacts varied by local industry, social context, and individual life histories. Lead authors Sarah Maguire and Ali Pourmond describe this as a rare collaboration between anthropology and isotope geochemistry aimed at illuminating lives during the industrial era. The study uses long bones and analyzes lead and strontium isotopes to distinguish lifelong exposure from contamination acquired after burial. To rule out post-burial signals, they compared bone signatures to burial soil at the Neptune Isotope Lab. This approach validates that the heavy metal signals reflect lifelong exposure rather than burial artifacts. The article, Geochemical Tales of Individual Lives in the Industrial Revolution, was published December 10, 2024, and involves researchers from multiple institutions, including the University of Miami, Ohio State University, Michigan State University, the Smithsonian Institution, Nashua Community College, Seton Hall University, and the Neptune Isotope Lab. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. We'll be back Monday with everything you need to know.